Though genetics are the source of many devastating disorders, they also hold the key to species betterment. In discussing the technological applications of DNA, it is important to remember that all cells begin as stem cells, which then acquire special characteristics to suit them to a certain function through the process of cell differentiation. This ability of stem cells means that they can be used to eliminate genetic diseases, regrow lost tissue, and battle degenerative diseases such as Parkinson's. Yet the uses of DNA do not stop there. It is possible to compare specific segments of DNA using restriction enzymes, which cut off the segment in question, and the gel electrophoresis process, which separates the segment to create a DNA fingerprint. These are an effective means to match crime suspects to evidence, as well as to determine an individual's parentage, because, other than twins, the odds of two individuals having identical DNA is vanishingly small. Useful as this is, it is not the most valuable application of restriction enzymes. Because they can isolate traits, they can be used to create recombinant DNA in transgenic organisms. Recombinant DNA is genetic material that has been taken from one organism as inserted into another, the transgenic organism. They are always sterile. The section of DNA cut with the restriction enzyme is placed in another organism's DNA by means of a vector. Plasmids, which are the circle of DNA in bacterial cells, are effective vectors because when the bacterial cells reproduce asexually, the success of the DNA transfer is apparent and that the isolated trait will be seen in both cells. Bacterial transfer has been used to treat diseases that involve a protein deficiency, such as diabetes and hemophilia, because the bacteria can be altered to create a protein, which is then given to those in need of it. Another type of transgenic organism is bug-resistant crops, which are made by adding a gene from the bacteria to the plant that is toxic to bugs. Finally, isolated genes can be used to combat genetic diseases through gene therapy, in which healthy traits are inserted into one's DNA to replace the mutated gene. Further progress is made in treating conditions this way through the Human Genome Project, which identified and mapped human genes. It enabled scientists to treat immunodeficiency and cystic fibrosis. However, not all results of DNA technology are positive. Genetically modified organisms, GMOs, while having eliminated wasteful water use and the need for pesticides, still have largely unknown effects on human consumers. Similarly, cloned crops lack genetic diversity by definition, making them susceptible to mass die-outs. Despite this, as DNA technology continues to progress, it will undoubtedly be a prominent figure in humanity's future.